Hello, Mr. Allman here, and this is your Allman Cast on our Enzyme Catalysis Lab. Previously in this unit, you learned about the enzyme lactase and how lactase is responsible for the decomposition of lactose, which is a milk sugar, into galactose and glucose. In this, you're also going to see a decomposition reaction that is also catalyzed by an enzyme. However, we're not going to be dealing with carbohydrates, we're going to be dealing with something that's actually toxic to living things. Every cell has an enzyme in it called catalase. And the responsibility of catalase is to decompose hydrogen peroxide. The same hydrogen peroxide that you might be familiar with from a pharmacy that's used to be able to kill bacteria and clean out a wound. This is also found in very small quantities in the mitochondria of our cells as a byproduct of respiration. Now, just as this is poisonous to bacteria in a cut, also hydrogen peroxide is toxic to you. So to have this build up in cells would really be problematic because it could cause a lot of damage. Hydrogen peroxide is a strong oxidizer, which means it's very reactive. So the hydrogen peroxide, as it moves around, might bond with and react with other proteins or possibly DNA in the cell, and as a result, the cell could die. In this lab, we're going to be using the breakdown of hydrogen peroxide by catalase as a way to be able to measure several different variables about the rate at which enzymes work. The catalase that you're going to get is going to be dissolved. It is water soluble, so you're going to have a solution in dropper bottles that has catalase in it. You will begin by adding some of the hydrogen peroxide, a specific amount, to a test tube. Remember that we'll be dealing with small quantities here. We'll have concentrated hydroxide and concentrated enzyme, so we don't need a large volume of liquid. So in doing so, you're going to use a cylinder, but remember, it's never a good lab practice to pour into a cylinder and then from that back into the bottle if you pour too much. So instead, make sure you use your dropper bottle so that when you aliquot your peroxide, you can take, using the dropper bottle, into your graduated cylinder, get the volume that you need, and then that can be poured into your test tube. One thing to remember about peroxide is that even though in our cells an enzyme is used to catalyze this decomposition, it will decompose on its own. So it's in this brown bottle because light is going to impact the rate at which this is broken down. So as this is exposed to light, some of that peroxide will on its own break down without an enzyme. So in terms of how this corresponds to your lab, it would not be a good idea to pour the peroxide into 10 different test tubes and use those test tubes over several different days. Even though it might be efficient to measure all of this out at one time, you only want to measure out the peroxide as you're using it, because as it sits here, it's going to bubble a little bit. And then you're affecting the starting concentration of peroxide in your test tubes. So avoid that, pour the peroxide as needed. Now, in this reaction, peroxide is going to be breaking down into two products. The first product is water, and the second product is oxygen gas. So you're going to get one of the oxygen atoms breaking away from the H2O2 to form oxygen gas O2. So in that reaction, we're producing water, a liquid, which is not easy to measure, but we are producing oxygen gas, which is easy to measure when we trap that gas in a container. So to do this, we'll be using a sensor that you should be familiar with from chemistry, which is the gas pressure sensor. The gas pressure sensor is going to the tip here is going to provide you a reading on your LabQuest in kilopascals of the pressure of whatever atmosphere it's exposed to. So in this case, the atmosphere of the air is around 98 kilopascals. So by attaching a tube to the end of the sensor, attaching a rubber stopper to the end of the tube, and now putting the stopper, which has a hole in the bottom, into the test tube, now whatever the pressure is inside this glass tube of the air that's trapped within there is being recorded on my LabQuest. So as the hydrogen peroxide decomposes here, it's going to produce oxygen gas. That oxygen gas will be added to the air above the tube, increasing the volume, increasing the number of molecules in a fixed volume. So as the number of molecules of gas increase here, that's going to cause an increase in pressure. So increases in pressure are going to give me a measurement of how much reaction has occurred. Now, because the LabQuest can record data over time, the increase in pressure over time can be used to determine the rate. I'll come back to that at the very end of the video. A couple of other logistical things to be aware of. One is to check your tube. So these tubes have been used a number of times for many different labs. So you need to check for any damage. Make sure that there's not any holes in the tube, particularly from burn marks, because if there is, then air will leak out and you will not see any rise in pressure. Similarly, make sure you check your connections at either end of the tube. And finally, when the stopper gets added in, you want to make sure that you push and wiggle, twist it into place to be sure that you get an airtight seal. When you perform the reaction, you're going to want to work together because there's three important things that need to happen. Your enzyme needs to be added, and remember, as soon as the enzyme is added, the enzyme is going to begin catalyzing the reaction and breaking down the peroxide and producing gas. So you want to be sure that you're ready to collect data. Don't add the enzyme first, only add the enzyme when you're ready to go. You'll add in the appropriate number of drops. Each drop, full drop, should be about 1 milliliter worth of liquid. Sorry, 1/10 of a milliliter of liquid. And then finally, secondly, you should then add the test tube stopper and start data collection by hitting the green arrow on your LabQuest. All of those things need to happen in very quick succession in order to increase the accuracy of your results. So if you delay in either putting the stopper on, or once the stopper's on, delay in hitting start on your LabQuest, that's going to delay the beginning of your collection of data, and your first few data points might not be able to compare as well to each other. So work together to be able to use many hands to be able to accomplish those three things: adding the enzyme, stopper, and LabQuest, all at the same time, or as close to the same time as possible. As it's going, remember that stirring does impact the decomposition because an enzyme needs to come in contact with the peroxide in order to do its job and catalyze the reaction. So you need to be consistent in terms of whether or not these are going to remain motionless and you're going to let it sit here, or if you're going to do any stirring, and if you're going to do any stirring, for how long will you stir it. Be consistent with that because the more you stir, the greater the increase in pressure you should see. Finally, in terms of analyzing data collection and collecting more data, remember that you have 3 channels into which gas pressure sensors can be added. So it may be beneficial to set up several different tubes, making sure that you label them, and collect data simultaneously. If that's something, it takes a little bit more to set up, but then it saves you a lot of time once that data collection has started. Finally, once the data is collected, you're going to want to use the LabQuest, once you have data here, to be able to determine for you what the slope of the line is going to be, because that's providing you with the rate. So you should see, as you look at a graph of the data, you should see that there is a line that is slowly or sometimes steeply increasing. So you want to be able to analyze, first find the linear fit that describes the slope of that line, because that slope, whatever that number is, that's giving you a number of kilopascals per second, that's providing you with a rate, and that's the number that you want to use to compare one trial to another. We'll be practicing this, you all using the same independent variable, but then you as a group will be choosing a new independent variable to be able to test, and you'll be conducting your own experiment based off of that independent variable choice and producing a lab report as a result. So please pay close attention to some of the details of the procedure, because this practice is going to be really important to ensure that you do this experiment correctly. As always, if you have questions during the lab, please let me know, or any questions before you begin that might be beneficial for the class, please ask. Thank you.